and welcome to Introduction to Clinical Research. My name is Marla Helly. I'll be your trainer today. Today's webinar, we're going to look at the following learning objectives. We're going to describe the different phases of drug development, as well as differentiating between both drug and medical device development. And then lastly, we're going to discuss the historical events that have occurred in helping us shape good clinical practice throughout the world in how we conduct clinical research. So before we get started, I think it's important just to look at the history of clinical research, and we'll also look at how clinical trials have come about, and also some of the things that have occurred in history that have not been so positive, that have helped shape how we now are protecting the human subject. I think what's interesting is you can actually look into the first clinical trial that was actually conducted, and this can be found in the Bible, actually, under the book of Daniel. There was an experiment that was conducted by King Nebuchadnezzar, who was also a military leader, and during his rule of Babylon, Nebuchadnezzar ordered his people who were in the military to consume a diet based upon meat and wine, because he believed that this would keep his military team in excellent health. However, there were several men who actually preferred to eat vegetables and objected. And so the king allowed these rebels or men to go ahead and follow a diet of legumes and water, and said, you can only do this for 10 days. And once this experiment ended, what he observed was those that had the diet, which was a vegetarian diet, over his meat eaters, appeared to be healthier. Now, we know that this wasn't a well-controlled study, but rather just looking at the overall beginning of how people were starting to say, let's do some comparisons. Let's see if there's a difference in how people perform or feel, or actually perhaps even might respond to a treatment. Later we see that there was a gentleman by the name of James Lind. He is a Royal Navy surgeon, and he is credited in determining what is the best method to prevent scurvy. And he did this looking at his sailors, and he said, we have a problem here with scurvy, and I want to evaluate how to best treat this. So he had 12 men with scurvy, and he said, I'm going to divide you up into pairs, and here is the regimen that he assigned. And he looked to see what regimen actually prevented the scurvy, which we know that scurvy is a condition where the gums bleed, and also we have teeth that are being lost. And he was hesitant at the beginning to use the oranges and lemons because they were expensive. However, what we discovered was this was actually beneficial, and this treated the scurvy. So this again kind of looks at some of the history. We also then look at how randomization and statistics come into play, and how we're now going to analyze our data. So in 1946, a medical research council of the United Kingdom just studied streptomycin and pulmonary tuberculosis. And a gentleman by the name of Sir Austin Bradford Hill developed randomization, which was a new method in which we're going to apply statistical design and processes and how we are going to eliminate bias. So he decided that we are going to randomly assign individuals either experiment 1 group 1 or experiment group number 2. And so the first group was assigned streptomycin and bed rest in the treatment of their pulmonary tuberculosis. And the second group was treated to bed rest alone. And this was done so there would be a random assignment to treatment. And what he observed was that those individuals that received both the antibiotic of streptomycin with bed rest actually had a response or an effective treatment to their tuberculosis versus those that were treated with bed rest alone.